Tonight we drift to the quiet edge of everything, into the black, breathless canvas between stars, where only the calmest, toughest travelers survive. In this darkness, there is no wind, no rain, no birdsong, no waves. Only the hush of space and the slow pulse of radiation older than memory. Against all reason, a few small beings from our blue world endure this silence. They do not rage, they rest. They fold into themselves like secrets. And then, when warmth returns, they open like morning. You are safe here. Let your shoulders loosen. Let your jaw soften. Imagine your body as a night ocean, its surface smoothing under moonlight. We will follow the gentlest of astronauts, creatures that teach us how to pause so completely that time slides past without harm. The water bear, whose patience is armor. Tiny crustaceans, carrying sleep like a spell. Quiet eggs of freshwater drifters, waiting out a universe, then hatching as if nothing happened. Space is an orchestra of extremes. A vacuum that whispers water from skin. Sunlight that strikes not as warmth, but as knives of ultraviolet light. A snowfall of invisible particles nibbling at life's alphabet. Out there ordinary bodies falter, but some have learned the ancient craft of becoming less. Motion for memory, heartbeat for hush, metabolism for a held breath, sugars and proteins knit into glassy veils. DNA tucked behind careful repair crews. Where we would fade, they bide their time. Sink with me gently, through clouds in the blue, till we rest just above the quiet earth. Picture a small capsule opening like a book to the void. Inside, tiny travelers lie asleep, some tucked into armored eggs, some folded into near crystals of themselves. The sun glances across them. The cold sharpens to frost. Weeks pass, months, a season, then air returns. A drop of water touches a shell. Life stirs again. No trumpet, no drama, just a sigh after a long, sweet pause. If this voyage helps you drift, you are welcome to curl up here again any night. And if you'd like more gentle science at bedtime, you can quietly subscribe or tap like. No rush, no noise, just a little lantern waiting for you next time. Tonight, we drift into a question both fragile and vast. What does it mean to survive in space? Not thrive, not walk, not breathe, easily beneath a blue sky, but to persist. Even when the very fabric of existence seems arranged against you, in the vacuum between heartbeats, when air is stripped away and silence presses from every direction, a few small lives hold secrets to endurance. Close your eyes now and imagine yourself suspended above the earth. Beneath you, oceans shimmer like pieces of spilled glass. Above you, stars pierce the velvet of night. Between them, nothing, the vacuum. No gentle wind, no molecules to carry sound. Just a silence so deep it becomes its own kind of sound, an endless stillness. Here in this void, the rules of life as we know it collapse. Let us step through them softly, one by one, like turning pages in an ancient cosmic book. When scientists say an organism survives in space, the meaning is delicate, layered. Do they mean the creature lived for minutes in vacuum, only to perish afterward? Or that it endured for months in frozen stillness, only to revive upon return to warmth and water? Do they mean active life, with cells dividing, feeding, breathing, or dormant life, paused, wrapped in protective stillness? This difference is crucial. For you, survival might mean waking tomorrow, lungs filling with oxygen, heart continuing its rhythm. But for certain tiny beings, survival is not waking at all, is pausing, suspending. It is the art of waiting, sometimes for years, sometimes for centuries, until the world turns kind again. So tonight, when we whisper survive, we will not mean walking proudly beneath alien suns. We will mean the spark of life does not go out, even when the candle is hidden. The most immediate killer in space is not cold, not radiation, but vacuum. On Earth, your skin holds in water because the atmosphere presses down, a quiet blanket of air. Space, that blanket is pulled away. Every droplet inside you wants to flee, to burst outward into the nothing. For a human, exposure to vacuum brings swelling, unconsciousness within 15 seconds, and death in minutes. Not because lungs explode, as myths sometimes say, but because oxygen slips out of blood, water evaporates from tissue, and the chemistry of life falters. For small, simple beings, the story is stranger. Some have found a way to let vacuum happen, to allow the water to leave. They dry into husks. Husks that still contain patterns of molecules arranged like a memory. Later, if touched by water again, those husks swell, stir, awaken. This drying without dying is called desiccation tolerance. It is the first secret of survival in space. If you stand in sunlight in orbit, your body could heat to boiling. If you drift into shadow, you could plunge colder than ice. The swing is savage. Plus 120 degrees Celsius in the sun, minus 150 degrees Celsius in shadow. Most life cannot handle these leaps. Cells rupture when water inside them expands into ice crystals. Proteins unravel like threads in fire. Yet some creatures use drying to protect themselves here too. In their dry forms, there is no water to freeze, no fluid to boil. Instead, sugars and proteins form glass-like cages around molecules, holding them still, safe, quiet. It is as if they wrap themselves in amber, soft, invisible amber made, not of tree sap, but of chemistry. The sun is both giver and destroyer. Its visible light warms Earth, powers photosynthesis, and paints our days with color. But beyond what your eyes see lies ultraviolet, a sharp sword of radiation. On Earth, we are shielded by atmosphere, by ozone, by clouds. In space, nothing stands in the way. UV light slices into DNA, breaking bonds, scattering instructions, tearing apart the script of life. For many creatures, vacuum alone is survivable. But vacuum plus direct sunlight is often the true killer. Survival rates plummet when UV exposure is added. Unless there is shielding, even a thin dust grain can be enough. And yet again, a few have learned repair. Some can stitch broken DNA back together as though reweaving a torn fabric. Others, in their dormant husks, endure simply because so much of their chemistry is frozen in stillness. Beyond sunlight, there are deeper storms. Particles hurled from the sun, or from galaxies far away, rain through space. These cosmic rays are like bullets, invisible, silent, but strong enough to break molecules apart. On Earth, magnetic fields and thick air protect us. In orbit, behind thin metal walls, astronauts still receive doses of radiation that, over years, could harm their health. For a tiny creature strapped outside a spacecraft, the bombardment is harsher still. Most cannot survive unshielded cosmic rays indefinitely. But again, some can repair. Some can simply endure, because in their dormant state, damage accumulates more slowly. Less immediately lethal, but still profound, is the absence of weight. Microgravity rearranges fluids in bodies, changes how cells signal, how muscles work, how bones stay strong. For creatures that survive in dormancy, this hazard matters less, for they do not actively live while floating. But for those carried into orbit while awake, worms, flies, even mice, microgravity reshapes biology in subtle ways. It teaches us that survival is not only about vacuum or radiation, it is also about living in a place where up and down no longer exist, where the body forgets how to feel its own weight. And here we arrive at one of nature's strangest gifts, cryptobiosis. The word means hidden life, describes states where metabolism slows so close to zero it cannot be measured. In this pause, time almost ceases. No food is eaten, no water flows, no breaths are taken, yet the potential for life remains. There are many flavors. Anhydrobiosis, life without water. Cryobiosis, life in deep cold. Anoxybiosis, life without oxygen. Osmobiosis, life in extreme salt. Each is like a lullaby written by evolution, a way of teaching cells to fold inward, to wait out disaster. When you think of survival in space, think of this. Not creatures marching boldly across the moon, but creatures curled inward, silent, glassy, waiting. They are not dead. They are not alive. They are in between. Scientists did not begin with humans when asking, can life endure space? They began with seeds, spores, microbes, and tiny animals. Outside space stations, platforms like Biopan, Expose, and Biorisk have opened and closed like drawers. Samples are placed within, dried, sealed, and then unlatched to open vacuum. For days, months, or years they sit bathed in sunlight or shadow. Later, they are brought back, rehydrated, tested. And again, to astonishment, some small sparks revive. Seeds sprout, microbes grow, tiny animals twitch awake. Not all survive. Often direct sunlight slashes their numbers. But the survivors whisper a truth. Life is tougher than we guessed. Take a deep, slow breath. Let it move gently in, gently out. Imagine that you too could curl into a shell, a husk, a safe waiting place, and let the storms of time pass over you. What does it mean that life can endure the vacuum of space? On the one hand, it is humbling. Humans, with all our technology, would perish in minutes outside the station walls. On the other hand, these tiny beings, so small you could sweep them from a pond with a teaspoon, can last months, even years. Fragility and strength are not opposites here. They're intertwined. The fragility of ordinary life leads to the invention of extraordinary strategies. The patience of waiting becomes stronger than the bravado of action. Perhaps there is a lesson for us in this, that survival is not always about force, but about stillness, about folding inward, guarding the spark, and waiting for gentler days. So now we know the battlefield. Vacuum, temperature swings, ultraviolet, cosmic rays, weightlessness. Against these hazards, most life falters quickly. But a few have mastered cryptobiosis, the art of survival through stillness. In the story to come, we will meet them. The water bear, the sea monkey, the rotifer, the worm, each a small keeper of secrets, each a teacher of patience. You begin. Let your breath soften once more. For tonight, survival is not a struggle. It is a rest. Imagine drifting gently into sleep, your body weightless, your thoughts dissolving into the dark. Somewhere in the silence between heartbeats, we step into the realm of one of nature's strangest wanderers. It is tiny, so tiny that 10 of them could curl together at the tip of a needle. Yet it carries within its soft, plump body a secret more astonishing than armor or fire, the ability to stand at the threshold of death itself and simply wait. This is the story of the tardigrade, the water bear, the moss piglet, the smallest astronaut who has ever pressed its nose against the airless door of space. First, picture it clearly. A body like a swollen, translucent pillow. 8 stubby legs, each tipped with little claws like grappling hooks. Its head a blunt snout, its mouth ringed with piercing stylets it uses to sip on algae cells or tiny invertebrates. Seen under the microscope, it waddles. It does not rush, it does not leap, it ambles. Its walk looks oddly like the slow, deliberate steps of a bear. Hence the nickname water bear. But do not mistake its softness for weakness. This creature, only 0.5 millimeter long, holds one of the most astonishing toolkits of endurance on Earth. Tardigrades live almost everywhere. In the thin film of water around moss, in puddles on rooftops, in the damp leaf litter of forests, in deep oceans, even at the frozen tops of mountains. Anywhere there is moisture, there may be tardigrades, plotting, grazing, carrying their secrets unnoticed. Their lives, when conditions are kind, are unremarkable. They eat, they molt their skins, they lay eggs. They live a few months, perhaps a year. But it is when the world grows harsh, when drought creeps in, when freezing grips, when air grows thin, that tardigrades reveal their second life. When danger looms, a tardigrade begins a ritual. Slowly it retracts its legs inward, tucking claws against the belly. Its round body shrinks and wrinkles. It releases most of the water inside itself, folding inward like a withering grape. What remains is a small, dry husk called a ton. In this state, metabolism slows to nearly nothing, less than 0.01% of normal. The creature neither eats nor breathes, becomes, in every ordinary sense, still. Yet the spark of life remains hidden inside, like a candle sealed in wax. Can ton form, tardigrades can survive. Complete vacuum, temperatures near absolute zero, heat above boiling, pressures greater than those at the bottom of oceans. Radiation doses hundreds of times what would kill a human. Decades of drought. It is as though they carry, in their humble bodies, a miniature version of eternity. When humans first dreamed of sending life into space, we chose mice, dogs, and chimps. Creatures large enough to resemble us, fragile enough to remind us of ourselves. But later, scientists turned to the tiny, And in 2007, tardigrades became the first animals to survive the open vacuum of space. Strapped to a European Space Agency satellite, they were launched into orbit. The hatch opened. For 10 long days, they were exposed to naked space, the vacuum, the swings of temperature, the ultraviolet glare of the sun. When the satellite returned, scientists rehydrated them. And to astonishment, many tardigrades stirred awake. Some even laid eggs that hatched into healthy young. It was the first proof that an animal, not merely a microbe, could endure the true unshielded void. The question is still being answered. But slowly, piece by piece, the puzzle emerges, drying as protection. By releasing water and folding into a ton, tardigrades prevent ice crystals from forming inside their cells during freezing, and avoid evaporation damage during vacuum exposure. Glass-like sugars inside the ton, special sugars like trehalose, form a glassy matrix, immobilizing proteins in DNA, keeping them from shattering. Unique proteins, the TDPs. Scientists discovered tardigrade-specific proteins, called TDPs, that can form a vitrified shield around cellular components. These proteins are like scaffolds that hold molecules still, protecting them from damage. DNA repair systems, even if radiation tears their genetic script. Tardigrades possess repair enzymes that can sew the fabric back together. Up the damage suppressor. One particularly strange protein named DSUP physically clings to DNA strands and shields them from radiation. When scientists inserted this gene into human cells, the cells gained remarkable resistance to X-rays. In short, tardigrades survive because they have transformed stillness into armor and chemistry into a spell of preservation. Of all hazards, radiation is most lethal in space. Yet tardigrades, though not immune, endure far more than expected. Some species withstand doses of ionizing radiation 1,000 times what a human could. They do not block radiation, they simply repair what breaks. Their DNA stitching enzymes work over time, guided by protective proteins. This suggests something extraordinary. Tardigrades did not evolve these abilities for space, evolved them on Earth, to endure cycles of drying and rehydration in mossy habitats. Radiation resistance may simply be a side effect, a bonus gift from evolution's cleverness. Why do we study tardigrades in such detail? Because they whisper of possibilities? Could their genes be borrowed one day to help protect human cells from radiation and spaceflight? Could their proteins be engineered into medicines that preserve vaccines without refrigeration? Could they teach us how to pause life safely, for organ storage, or even for long journeys between stars? Already DSO protein has been tested in labs. Already TDP-like mechanisms are being explored for biotechnology. Tardigrades are no longer just curiosities of moss. They are research partners. Close your eyes for a moment. Picture a tardigrade, curled into a ton, motionless, dry. It looks lifeless, yet inside, there is stillness so deep that it cradles survival. Now imagine yourself in that stillness. No rushing thoughts, no urgent movements, just waiting, resting. Trusting that life can be carried quietly even through deserts of time. Some scientists wonder if tardigrades can endure years in space, drifting dormant. Could they hitchhike on dust between planets? Could they, in theory, become messengers of life across the solar system? Most say the odds are low. Radiation, over long spans, would still destroy them. And yet, the fact that the question can even be asked reveals how extraordinary their endurance is. Tardigrades stand at the airless door, knocking gently. They remind us that life is not as fragile as we once believed, that the spark can endure silence, vacuum, and even death's shadow. At first glance, tardigrades are plump and harmless. They look like plush toys under a microscope. Yet within them lies one of the fiercest arsenals of survival ever discovered. Perhaps that is the lesson, that strength is not always hard, sharp, or violent. Sometimes it is soft, patient, slow. Sometimes survival comes not from resisting the storm, but from folding inward and waiting until the storm has passed.
Ow.
The water bear teaches us that life's endurance is not always a fight. Sometimes it is a pause. And so, as you drift deeper into tonight's rest, remember the water bears. Tiny, plump, slow-walking creatures, invisible to the naked eye, yet capable of what we cannot, touching the vacuum of space and returning alive. They are the smallest astronauts, the keepers of stillness, the teachers of patience. And in their story, perhaps lies a gentle lullaby. That survival, even in the harshest night, is not impossible. It is only waiting. Settle in, soften your breathing, and let your mind drift like a tide rolling across the shore. Tonight we wander into shallow saltwater pools, into places where nothing seems permanent, where lakes appear and vanish, where life itself teeters on the knife's edge of existence. Here in the shimmer of evaporating waters lives a creature both fragile and unstoppable. Not majestic, not intimidating, but humble, transparent, delicate, the brine shrimp. You may know it by another name, the so-called sea monkey, sold in tiny packets to children as living toys, a novelty, a curiosity, a creature summoned to life with nothing more than a splash of water. Yet behind that cheap plastic aquarium lies one of biology's most extraordinary secrets, a survival strategy so powerful it blurs the line between life and death. A pause button written directly into the code of existence. At first glance, there is nothing astonishing about the brine shrimp. Its body is slim, segmented, transparent. Its head carries stalked eyes and a crown of feathery limbs it uses for swimming. It flutters through salt water with a motion like flying underwater. Its wings beat rhythmically. Its tiny heart pumps in plain view through its glassy chest. It feeds on microscopic algae, filtering green specks from the water with a slow, gentle grace. And yet its beauty is subtle. In ponds, salt flats, or inland seas too harsh for fish, brine shrimp drift in the millions, turning waters pinkish with their swarms. They are food for flamingos and gulls. They are not predators, not conquerors, just humble plankton, sustaining others. But brine shrimp possess a secret far beyond their fragile appearance. To understand their magic, we must step into their world. Brine shrimp do not live in ordinary rivers or lakes. They dwell in extremes. Salt lakes, like the Great Salt Lake in Utah, or Mono Lake in California, or remote saline pools across Asia and Africa, are their havens. These are waters too salty for fish, too unstable for most creatures. Rain may swell these lakes one year, then drought shrinks them the next. Temperatures swing wildly. Water may vanish altogether, leaving cracked earth only to return seasons later. To most life, such environments are death traps. But for brine shrimp, they are opportunities, because they carry within them a time capsule. When conditions turn deadly, when water evaporates, salinity rises, or winter closes in, female brine shrimp produce not ordinary eggs, but cysts. A cyst is not merely an egg. It is an embryo sealed in stasis. Its outer shell is tough, almost indestructible, like biological armor. Inside, the embryo halts its growth, slipping into a state called cryptobiosis. In this state, metabolism ceases. As no energy is burned, no oxygen consumed, biochemistry freezes. Enzymes normally delicate are immobilized, yet preserved. Water is expelled. The embryo dries almost completely, preventing ice from rupturing cells. DNA is shielded, damage is slowed, mutations resisted. It is as though life has been folded into silence, waiting patiently for the world to soften again. These cysts can endure extremes that should be impossible. Decades without water, freezing below -190 degrees Celsius, colder than liquid nitrogen, boiling heat, near vacuum resembling space itself, radiation doses that shred molecules. And then when water returns, when salt balances, when warmth touches, the cyst awakens. Cells stir, growth resumes. And a new brine shrimp hatches as if no time had passed at all. This is why brine shrimp became sea monkeys. In children's toy kits, packets contain dormant cysts, dry and lifeless looking. Add water, and within hours they awaken and hatch, swimming like tiny aliens in a plastic tank. To a child's eyes, it is magic. To a scientist's, it is the purest example of cryptobiosis in action. Life paused, time defied, survival ensured. But how does a cyst truly stop time? Inside the embryo replaces water with special sugars. Trehalose, again the same sugar used by tardigrades. Trehalose stabilizes proteins and membranes, locking them in place like glass. Even more fascinating, brine shrimp embryos produce unique proteins called Li proteins. Late embryogenesis, abundant proteins. These proteins act as molecular sponges, binding to cell components and preventing collapse during drying. Lee proteins and tree hollows together create a microscopic glassy state. Everything essential is preserved exactly as it is, frozen not by cold, but by chemistry. This is why cysts can last not years, but centuries. Some revived cysts have been dated to nearly 100 years old. There is speculation, though still unproven, that some cysts might remain viable for thousands. Scientists, intrigued by this endurance, have hurled cysts into conditions few organisms ever meet. Cysts have been frozen in liquid helium, heated close to boiling, exposed to harsh chemicals, even blasted with space-like radiation. Each time many die, but many survive, and with just a drop of water, they return. In one extraordinary case, brine shrimp cysts survived direct exposure to space aboard satellites, much like tardigrades. They endured vacuum and cosmic rays, then hatched when brought back to Earth. They are not immortal, but their patience stretches further than almost any other visible creature. What is a cyst, if not a seed? Plants scatter seeds to endure winters or fires. Some seeds remain dormant for centuries in desert soils. Brine shrimp cysts are the animal counterpart, a seed of flesh, a pause in the dance of living. Perhaps that is why they feel so mesmerizing. They embody the possibility that life need not flow like a river, but can be stopped like a clock and restarted when the moment is right. Imagine it. To pause completely. To fold into silence. To let decades pass over you like falling sand, without hunger, without pain. Brine shrimp embryos do not struggle through the drought. They do not resist. They simply wait. And perhaps that, too, is a lesson for us. That sometimes survival does not mean constant effort, but trust in stillness. Scientists have even borrowed lessons from brine shrimp cysts for human technology. Medicine storage. Could vaccines or blood products be dried and stored without refrigeration, preserved by trehalose Lee proteins? Brine shrimp suggests yes. Space travel. Could cryptobiosis one day help humans suspend metabolism for interstellar journeys? Science fiction imagines it. Biology hints it may not be impossible. Agriculture and biotechnology. Lee proteins are now engineered into crops to improve drought resistance. What brine shrimp use for survival, humans may adopt to feed the future. The humble sea monkey, once a child's pet, is now a teacher of biotechnology. Close your eyes again. Picture a tiny cyst, dry and unremarkable, rolling across a salt flat in the wind. It looks like dust. It could be overlooked as nothing. But inside, curled in silence, is the memory of life. Waiting. Waiting for rain. Waiting for time. When conditions soften, it will open, and what seemed dead will prove alive. The story of the brine shrimp reminds us that survival does not always roar. Sometimes it whispers. Sometimes it withdraws. Not through conquest, but through patience. Not through force, but through trust in time's return. In their cysts, brine shrimp carry one of nature's oldest lessons, that even when the world dries and cracks, when everything around you vanishes, life can find a way to pause and to resume. So as you drift into tonight's sleep, let the thought settle gently, that even in silence, even in stillness, even in what seems like endings, there can be beginnings folded within. The brine shrimp's secret is not immortality, it is patience. The art of waiting for the right tide. And maybe, as you rest, you can borrow a little of that art. To trust the pauses. To let time hold you gently. To believe that renewal always waits with the next drop of water, the next dawn, the next breath. Settle deeper into your pillow. Breathe slowly. Imagine you are standing at the edge of a quiet lake, the water stretching still and silver beneath the evening sky. Dragonflies skim its mirrored surface. Somewhere far below, unseen, a secret library lies waiting. Not a library of stone or shelves, not a library filled with paper and ink. This library is written in eggs, tiny capsules of life resting in the mud. Pages not of words, but of DNA. Volumes not of history, but of possibility. This is the story of Daphnia, the water flea. And its most astonishing gift, resting eggs that outlast centuries. Eggs that store the memory of lakes. Eggs that function as a genetic archive, a biological time machine. At first glance, Daphnia hardly looks like a creature that could carry such grandeur. They are tiny crustaceans, just a few millimeters long. Under a magnifying glass, their bodies are transparent domes, within which you can see every detail. A beating heart, rhythmic with the soft pulsing of life, digestive tracts. Eggs nestled within the brood chamber. Their antennae are long and feathery, which they use as oars to row themselves through the water in jerky but graceful movements. Despite their miniature size, they are pillars of freshwater ecosystems. Daphnia feed on algae, bacteria, and tiny detritus, keeping waters clear. They are prey for fish and insects, forming a vital link in food webs. If lakes are living systems, Daphnia are their quiet engineers, regulating balance without ever being seen. But like all fragile things, their survival depends on ingenuity. And in the unpredictable, shifting world of freshwater, they have perfected one of life's most ingenious strategies: the resting egg. Lakes may look timeless to human eyes, but they are ever-changing. Rain and drought alter their depths. Winters freeze their surfaces. Summers warm their shallows. Algae blooms rise, then crash. Oxygen levels fall and climb. For a creature like Daphnia, which lives in open water vulnerable to temperature, oxygen, and predators, every year brings new dangers. A fish invasion may sweep through a lake, devouring the population. A sudden freeze or drought may collapse conditions entirely. To survive such cycles, Daphnia have two modes of reproduction. In good times, they clone themselves rapidly by parthenogenesis. No males required, just endless self-copying to flood the water with daughters. This allows them to bloom quickly when conditions are safe. But when the lake becomes harsh, when winter approaches, food dwindles or predators loom, they shift strategies. Males are produced, mating occurs, and the females create something remarkable: resting eggs, also known as amphibia. Unlike ordinary eggs, resting eggs are encased in a protective shell. It's often dark and opaque, carried in a saddle-shaped case called an amphibium. These capsules sink to the bottom of the lake where they wait. Inside, life slows almost to nothing. The embryo halts its development, just like the cyst of a brine shrimp, entering a dormant state that can last astonishingly long. But while brine shrimp cysts are remarkable for resisting extremes, Daphnia's resting eggs have another role. They function as a record, genetic storage of entire populations across centuries. Some amphibia lie buried for hundreds of years in sediment. Yet when scientists collect them, clean them, and expose them to the right conditions, many still hatch. Out of mud that remembers wars, plagues, and revolutions, tiny transparent creatures emerge, alive as though summoned from history. This ability has given rise to a field called resurrection ecology. Scientists take cores of lake sediments, extracting layers like the rings of a tree. Each layer contains amphibia from a particular era, preserved where they sank. By hatching these eggs, researchers can literally revive populations from the past. It is as though one could plant a seed from ancient soil and watch a long-extinct forest return. Through this method, scientists have reconstructed centuries of ecological history. They have observed how Daphnia populations adapted to rising pollution during the Industrial Revolution, how they responded to pesticide runoffs from farming, how they endured acid rain, warming waters, invasive predators. Each layer of mud is a time capsule. Each egg is a page in the lake's autobiography. Pause and let that thought sink in. Beneath the still surface of a lake lies a living archive, a biological library where every generation leaves behind sealed letters to the future. It is not a memory we can read with ink. We must coax it alive, watching embryos stretch their limbs, beating their tiny hearts again after centuries of silence. In that act, the past does not just speak, it swims. This makes Daphnia resting eggs unlike any other survival capsule. They are not only tools of endurance, but storytellers of entire ecosystems, narrating how lakes have shifted across time.
Wow.
How do these eggs survive? Their shells are remarkably resistant, shielding them from crushing sediment, bacterial attack, and chemical corrosion. Inside, metabolic activity drops to nearly zero. Protective proteins stabilize DNA. Antioxidants ward off damage. The key, once again, is cryptobiosis, a suspension between life and death. Unlike fragile adults, which live mere weeks, the embryos inside Aphippia may remain viable for centuries. It is a strange paradox. The most delicate creatures create the most durable offspring. A tiny water flea, ephemeral in its own life, leaves behind eggs that can wait patiently longer than dynasties, longer than empires. In laboratory experiments, resting eggs collected from sediments 2 or 3 centuries old have been hatched successfully. Imagine. Creatures emerging from eggs older than electricity, older than railroads, older than photography. Some attempts push further. Scientists have tried to hatch Aphippia dating back more than 500 years. Results are mixed, some awaken, others fail. But even those that do not hatch still hold genetic information. By sequencing their DNA, researchers can reconstruct the genetic composition of ancient populations. Even when life itself does not revive, it is as if the library has 2 ways of speaking. Some volumes can be reopened and read directly. Others can only be translated through fragments, yet both preserve the story. The resting eggs do not only tell us of the past, they hint at the future. By comparing ancient and modern Daphnia, scientists observe how populations have gained or lost resistance to toxins, diseases, or predators. This reveals the evolutionary capacity of small creatures to adapt quickly, and also their limits. In a warming world, with human-driven change, reshaping lakes at unprecedented speeds, eggs are a resource of hope and warning. They show us how life bends before pressures, how it learns, but also when it breaks. Perhaps these tiny crustaceans are guardians, carrying not just their own survival, but the lessons we need to safeguard freshwater itself. Now, let us imagine ourselves drifting at the bottom of a lake, weightless in the dark mud. Around us lie countless Aphippia, scattered like black seeds, like shelved books in an unseen library. Some hold embryos from decades ago, some from centuries. Each one is a sealed moment in time, heartbeat captured and paused, a tiny flicker of life waiting for the call to return. In this silence, the concept of time feels different. Human years are loud, rushing, filled with noise and urgency. But here, time is sediment. Slow, steady, layering memories grain by grain. The eggs wait without impatience. They do not know days or centuries, only know readiness. When you step back, you see a pattern. Seeds in deserts, spores in fungi, cysts in brine shrimp, eggs in Daphnia. All life carries strategies of waiting. Each one is an answer to the unpredictability of the world. Each one is a quiet defiance, a refusal to end simply because the present is hostile. Resting eggs of Daphnia are one of the most elegant manifestations of this truth. They are not only survival mechanisms, they are continuity itself. We too long for such continuity. Archive books, photos, digital files, time capsules, even frozen seeds in vaults in the Arctic. Bury treasures for future generations, hoping someone will open them and understand who we were. The lakes have been doing this for millennia without our help. Their archives are deeper, quieter, and infinitely more patient. Perhaps in a sense, the Daphnia are librarians. Their eggs are books bound in chitin, written in DNA, cataloged in sediment. The library needs no caretakers. It tends itself, writing volume after volume, generation after generation. And when a scientist's hand dips into the mud and revives an ancient egg, it is like lifting a book from a forgotten shelf and finding the words still fresh, the story still alive. As you rest tonight, imagine the lake once more, its surface calm under moonlight, its depths filled with shadows and silence. Library vast and unseen, where resting eggs sleep patiently. They remind us that memory does not always need ink, that history is not always carved in stone. Sometimes, history is alive, sealed in capsules, waiting to be called back. Perhaps your own mind, as you drift into dreams, is not so different. Thoughts pause, reshape, and sometimes return with fresh life. Memory itself is a kind of egg, delicate, enduring, carried forward through the layers of time. So breathe softly, trust the stillness. Somewhere in the mud of your own being, entire libraries wait to awaken when the moment is right. Tonight, we drift closer to the forgotten currents of life's smallest wheels. Rotifers, tiny creatures, so delicate and so persistent that even the name carries a whisper of spinning and flowing. Rotifer wheel, fur forebearer? Imagine for a moment that the universe itself holds a quiet ribbon winding endlessly forward, sometimes unraveling, sometimes mending, and along this ribbon, little lives cling with astonishing patience. Among these lives are the rotifers, nearly invisible, smaller than the width of a grain of sand, and yet among the most durable travelers of Earth's long history. They are called wheel animals because at their head is a crown of cilia that turn like the blades of a watermill. To a passing eye under the microscope, they appear to spin endlessly, drawing currents of water, sweeping invisible algae and bacteria toward a hidden mouth. And yet, this is only the beginning of their story. For beneath their simplicity lies one of nature's most remarkable survival toolkits. One that has carried them not just through drying ponds and frozen lakes, but even into the silent emptiness of space itself. Rotifers belong to a lineage that is more than 35 million years old, possibly much older. Among them, the beetaloid rotifers are the most studied, and they hold a secret that unsettled evolutionary biologists for decades. They appear to have abandoned sexual reproduction entirely, while almost all animals reshuffle genes through mating. Beetaloids have persisted without it, copying themselves generation after generation for millions upon millions of years. And yet, instead of collapsing into genetic decay, they have thrived. How one answer lies in their ability to repair broken DNA, a talent honed by the stresses of their environment. When they dry out completely, a state called anhydrobiosis, their cells fracture. Their DNA strands shatter into dozens of pieces. To almost any other animal, this would mean death. But beetaloids stitch themselves back together again when water returns, carefully repairing, replacing, and sometimes borrowing DNA from the world around them. Studies have shown traces of horizontal gene transfer in their genomes, genes from bacteria, fungi, even plants, woven into their own code. They are mosaics of survival, stitched quilts of life itself. In this strange strategy lies their resilience, their ability to endure not only the ordinary harshness of Earth, but also the extremes of outer space. Not long ago, betyloid rotifers were selected for spaceflight studies aboard the International Space Station ISS. Scientists wanted to know, could these creatures, already masters of desiccation and DNA repair, endure the stresses of orbit? Could they survive microgravity, cosmic radiation, and, when exposed, perhaps even the raw vacuum of space itself? The results were startling. When desiccated and launched into orbit, rotifers revived with strong survival rates. Transcriptomic studies, mapping which genes were switched on, revealed a deep stress response wiring. It was as though rotifers had rehearsed this play many times before, even if space itself was new to them. Their systems for protecting proteins, repairing DNA, and scavenging free radicals leaped into action. Some experiments placed them outside protective shields, where radiation and cosmic rays could reach them more freely. While their numbers diminished under these harsher exposures, a core of survivors endured. This resilience echoed their earthbound ability to tolerate extreme UV and ionizing radiation, an ability almost unimaginable for animals of such small size. Here, then, was evidence. Rotifers might not float about in the void forever, but dried into dormancy, they could certainly endure the kind of silence that space imposes. When we picture life in space, we imagine great vessels, domes, and machines. But life's first spacecraft has always been its own body. The rotifer, in its amphibia-like casing, or its dried state, becomes a vessel of patience. To drift in orbit for days or weeks is not unlike waiting at the bottom of a frozen pond, or sealed inside a mud-flaked egg, enduring drought. The rotifer does not rage against the silence. It yields to it. It pauses. Its ribbon of life coils in on itself, still waiting. And when the moment is right, when a drop of water returns or when conditions soften, the wheel turns again and life continues. There is a kind of cosmic metaphor in this. Survival, not by force, but by stillness, by waiting out the storm, by trusting that endurance itself is a kind of movement, a passage through time. Take a deep, slow breath and imagine those tiny wheels turning gently in the waters of your own thoughts. Exhale slowly and let that wheel become still, waiting like the rotifer, patient in its silence. Though rotifers are astonishingly hardy, they are not invincible. They cannot remain indefinitely exposed to raw cosmic rays without protection. The ultraviolet light of the unfiltered sun slices through DNA relentlessly, and only in brief or shielded exposures can they persist. Like tardigrades and brine shrimp, they survive space not by living actively, but by embracing dormancy, letting go of activity, letting go of time itself. This is important to remember, for it shifts our imagination away from fantasies of immortal spacefaring creatures and back toward the truth. Survival in the cosmos is always conditional, always a negotiation between fragility and patience. And yet, what beauty there is in that negotiation. What elegance in the way such a tiny being can teach us about resilience, about silence, about letting stillness itself be the path through danger. Scientists studying rotifers have called them models for DNA repair, stress resistance, and even aging research. The fact that they survive such intense damage and then rebuild themselves is a living inspiration for biotechnology and medicine. In the hush of their microscopic world lies a blueprint: how to accept damage without despair, how to knit the fragments of life back together, how to borrow wisdom from neighbors when needed. Is this not a lesson for us as well? That we too are capable of repair, even after shattering? That silence does not mean the end, but perhaps the beginning of renewal. The Rotifer's ribbon carries not only its own story, but echoes of ours as well. As we move deeper into our exploration of spacefaring animals, let the Rotifers linger in your imagination. Picture them, thousands of them, each smaller than a fleck of dust, quietly drying into stillness, waiting, floating. They need no spaceship, no suit, no food, no water, only time and the patience to endure it. It is a kind of poetry that such fragile creatures can do what humans cannot: step briefly into the airless dark and return alive. And yet, we should not envy them. Their survival is not a victory march, but a lullaby, a soft folding of life into silence, a waiting for the wheel to turn once more. Inhale gently, as though drawing in the ribbon of a dream, and exhale, letting that ribbon unwind, flowing outward into the dark, carrying you deeper into calm. The rotifers remind us that not all survival is loud. Some survival is quiet, unassuming, and ancient. A persistence of patience, a resilience of repair. In the stillness of space, the wheelbearers continue their turning, invisible but enduring, spinning their ribbons of life across time. And as you rest tonight, drifting into your own inner silence, may you feel that same quiet resilience inside yourself. The ability to pause, to repair, to continue, softly, patiently, endlessly. The stars above us burn with ancient fire. But below, hidden in the soil, the sand, the roots of grass, the lining of intestines, and the silent chambers of melting ice, live creatures so common, so persistent, that they might be called the true keepers of Earth. They are nematodes, worms though, not the kind that wriggle heavily on sidewalks after rain. These are finer, thinner, subtler worms. Threads of life coiled through nearly every ecosystem, from mountaintops to ocean trenches, from compost heaps to your very eyelashes. If Earth's biomass were weighed, nematodes would tip the scale more heavily than most animals combined. By number, they may represent 4 out of every 5 animals alive today. And among these countless multitudes lies a story not just of persistence, but of cosmic patience. For nematodes have shown us that life's thread can endure in silence for astonishing lengths of time, frozen, dried, drifting through space itself. Their ribbon of survival stretches not only across the face of the planet, but also into the vast experiment of space. In 2018, scientists announced something extraordinary. From samples of Siberian permafrost, soil that had lain frozen for tens of thousands of years, 2 species of nematodes stirred back to life. One, Panagrolomus calimicis, the other Plectus murai. Both had been entombed since the Pleistocene, more than 40,000 years ago, when mammoths walked the tundra. In the deep freeze of the Earth's own natural cryogenic vault, these worms had endured without water, without air, without motion. Their cellular metabolism had slowed into a glassy stasis, a suspension not of minutes or days, but of millennia. And when the ice finally softened, when human scientists gently coaxed them into warmth, they twitched. They lived. After 40 millennia, the ribbon of their life unfurled again, continuing as if no time had passed. What does it mean that such a simple animal can pause for so long? That a body no thicker than a hair can hold within it the patience of ages? To contemplate this is to feel time loosen, to feel history itself become a kind of dream, where even the coldest silence cannot erase life's hidden spark. Take a slow inhale. Imagine drawing in the cold, crisp air of frozen ages. Exhale gently and feel the thaw, the quiet return of motion after stillness. Nematodes have not only endured the frozen ground, they have also traveled beyond it. In 2003, the Space Shuttle Columbia suffered its tragic disintegration on re-entry. And yet, when debris was examined, a living cargo was found among the wreckage. Nematodes, tiny roundworms of the species Canahabditis elegans, shipped as model organisms for research, had survived the fiery destruction of the shuttle. It was a sobering reminder that life, at its most modest, can endure shocks that no spacecraft can protect us from. The worms, shielded in simple canisters, lived through fire, fall, and chaos. Later on the International Space Station, nematodes were studied to understand how microgravity affects muscles, genes, and longevity. Because their biology mirrors ours in miniature, what happens to them teaches us about what happens to astronauts. They shrank and stretched in microgravity, their muscles weakening, their cells adapting. They became not only passengers, but participants in humanity's search for survival in space. And in each case, the lesson was clear. Resilience is not measured by size or by strength, but by adaptability. What gives nematodes this power? Like tardigrades, nematodes can enter cryptobiosis, a state where metabolism grinds to nearly nothing. In the absence of water, they dry themselves into a tiny husk. Sugars like trehalose replace the water in their cells, stabilizing membranes and proteins. Their DNA, too, can break during the long freeze, but nematodes have refined repair mechanisms that carefully stitch it back together. Some nematodes resist radiation better than most insects. Others tolerate extremes of pH, salinity, or pressure. In the quiet theater of soil and ice, they rehearse the very same dramas that await in orbit and beyond. To live as a nematode is not to dominate, but to persist, to curl into stillness when the world becomes unbearable, and to wait until the world softens again. There is something profoundly humbling about these worms. For all our rockets and spacesuits, for all our calculations and shields, humans struggle to endure where nematodes wait patiently. They need no food stores, no architecture of steel, only the ancient chemistry of cryptobiosis. Imagine, then, the possibility. A dry husk of a nematode, sealed in the crevice of a meteorite blasted into the sky. Could it endure the vacuum, the cold, the radiation? Could it drift for years before landing on another body, and there, in the presence of water, revive? We do not know. But the permafrost worms whisper that it may be so. That life's ribbon is not fragile thread, but braided cord, capable of spanning more than one world. Nematodes have been humanity's teachers for decades. Canahabditis elegans, in particular, has served as a model organism in laboratories worldwide. Its genome is mapped, its cells tracked from embryo to adult, every division observed. Through them, we learned about apoptosis-programmed cell death, about nervous systems, about how genes choreograph life's dance. And now, as they journey to space with us, they continue their lesson. That survival is often a matter of letting go, of entering stillness, of trusting the hidden resilience of the body. When astronauts return from orbit, weakened by microgravity, they are not so unlike nematodes, revived from desiccation, fragile but alive, hurting but capable of repair. So tonight, as you drift deeper into rest, let the nematodes coil gently in your imagination. Picture them waiting, whether in the icy soil of Siberia, in the capsule of a spacecraft, or in the dark between stars. They are not heroes or giants. They are not fierce. They are simply patient. And in their patience lies a lesson we can carry into sleep. That survival does not always demand motion. Sometimes it asks for surrender, for stillness, for trust in the quiet. Inhale slowly as though drawing in the hidden patience of the worms. Exhale gently, letting your body coil softly into rest, safe and enduring. The nematodes, the worms that worms at worlds, are a reminder that life's thread is stronger than it seems. Through fire, ice, vacuum, and time itself, they persist. And so can you. As you drift into sleep, know that your body, too, holds the same quiet resilience. Like the nematodes, you can weather the silences of life and awaken again, whole. When the world becomes unbearable, too dry, too cold, too hot, too empty, most living things falter. Muscles stop moving, cells collapse, water evaporates, and life comes undone. But there exists, hidden in the small and the overlooked, a rule as old as Earth itself. When you cannot go on, you do not need to perish. You can simply stay still. This is the law of cryptobiosis, a survival script written deep into the genomes of some of life's humblest survivors. It is not death, not sleep, not hibernation. It is closer to vanishing, like a flame blown out, yet leaving behind a spark. A living suspension where time itself seems to lose meaning. Cryptobiosis is not the exception in nature. It is a quiet secret shared across kingdoms of life. In water bears and rotifers, nematodes, and brine shrimp, even in the dormant embryos of plants and microbes. Each of them, in their own way, has mastered the same paradox: to survive by surrendering. To understand cryptobiosis, picture a pond under the summer sun. Its water evaporates, turning mud into dust. Fish suffocate, insects vanish. But in that dust, a brine shrimp cyst curls tighter, coated in layers of protection. Its metabolism slows not just by half or 1/100th, but to something so close to zero it can scarcely be measured. Or picture an Arctic tundra, winter pressing hard, temperatures plunging to -60 degrees Celsius. Beneath the soil, a rotifer lies twisted into a microscopic knot. Its body is desiccated, its cells glassy. It waits. Snow covers the ground for years, perhaps decades. Yet at the first touch of thaw, a drop of water enters. Molecules shift, and the rotifer uncoils, alive again. Life without cryptobiosis is fragile. Life with it is timeless. What happens inside these creatures when they surrender to cryptobiosis? The secret lies in biochemical substitution. When water is drawn out, whether by freezing, drying, or vacuum, the cell risks collapse. Proteins denature, membranes tear, DNA fractures. To counter this, organisms deploy a remarkable trick. Sugars, especially trehalose, flood the cells, replacing water molecules in their structures. Trehalose forms a glass-like matrix, stabilizing proteins and lipids in a solid, immovable embrace. Instead of shattering, the cell freezes in order, preserved in a biological amber. Some tardigrades employ a different strategy, producing special proteins called TDPs, tardigrade-specific intrinsically disordered proteins. These do not fold into neat structures like most proteins. Instead, they remain flexible, forming a gel-like shield that keeps molecules stable during desiccation. Radiation, too, is countered, not by defiance, but by patience. DNA may indeed shatter into pieces, but once revival begins, the organism slowly, meticulously repairs the damage. A body fragmented by cosmic rays can weave itself whole again. It is not strength in the usual sense. It is faith in repair, faith in stillness. In cryptobiosis, time loses its grip. A tardigrade desiccated for a decade can revive. Brine shrimp cysts have hatched after nearly a century of dry storage. Nematodes from Siberian permafrost awoke after tens of thousands of years. Spores of bacteria, locked in salt crystals, may have endured for millions. Imagine, for all the while that civilizations rise and fall, glaciers advance and retreat, stars wheel across the heavens, a tiny husk of life lies dormant. Not aging, not changing, not aware, just waiting for water. This is not immortality. When revived, the organism continues aging as if no time has passed. But the interlude is erased, skipped, like a page never written. Cryptobiosis is the closest life comes to time travel. Cryptobiosis is not a single state, but many, each adapted to a different threat. Anhydrobiosis, survival against drying. Creatures lose nearly all water and replace it with sugars or protective proteins. Cryobiosis, survival against freezing. Metabolism halts. Ice crystals are prevented from forming. tissues preserved like glass. Osmobiosis, survival against extreme salinity. Organisms shut down, resisting the pull of salt crystals. Chaosis, survival against oxygen absence. Bodies enter paralysis, waiting until oxygen returns. Each pathway is like a different door into silence, opened only when conditions demand it. Together, they form a handbook for surviving the impossible. Why does cryptobiosis matter beyond Earth? Because the universe is filled with deserts, vacuums, extremes of heat and cold. For any form of life to travel beyond its birthplace, it must learn to endure them. Experiments have shown that organisms in cryptobiosis can survive exposure to outer space. Tardigrades dried into husks endured the vacuum and radiation of low Earth orbit. Nematodes survived re-entry aboard Columbia. Seeds carried across decades and journeys germinated still. It is possible, then, that cryptobiosis is not just a quirk of Earth, but a bridge for life between worlds. A cyst, a spore, a desiccated husk drifting through the void may one day revive when it encounters warmth and water. In this way, cryptobiosis may be the true carrier of Panspermia. The idea that life spreads through the cosmos not as thriving animals or plants, but as sleepers waiting to be awakened. Why does this matter for us here, listening in the quiet of night? Because cryptobiosis, though far beyond human ability, mirrors something we practice every day: sleep. When we rest, our metabolism slows, our consciousness dissolves. Our body repairs itself, cleanses toxins, rebalances chemistry. We surrender awareness in order to continue. In our beds each night, we practice a miniature version of the same rule, to survive by stillness. And like cryptobiosis, sleep erases time. Ours vanish in an instant. One breath, one blink, and suddenly it is morning. When you drift into sleep tonight, you too will step into your own soft cryptobiosis, a state not of death, but of pause, a thread of life held steady until dawn's return. There is a quiet wisdom in these tiny creatures. Their lesson is simple. You do not need to endure every moment actively. When the world is too harsh, you may step aside. When time is too heavy, you may let it pass. Survival is not always a fight. Sometimes it is a pause, curl inward, refusal to be broken by waiting. And in this patience lies resilience greater than strength. Cryptobiosis is nature's reminder that survival does not always roar. Sometimes it whispers. Sometimes it folds itself inward and waits in silence. As you drift now into your own night of rest, imagine yourself like those ancient sleepers, tardigrades, nematodes, rotifers, curled safely into stillness, untouched by time. When morning comes, you will stir again, whole. Life has written into its code this secret, the rule of staying still. And tonight, that rule belongs to you too. There is a door in orbit that few have seen open, not the great hatch that astronauts pass through when leaving for a spacewalk. Not the inner doors of modules where experiments hum and computers glow. This is a smaller door, secret door. It opens not for people, but for creatures too small to notice. For seeds, for spores, for lichens clinging to stone, for dried husks of water bears. It is the door to the outside platforms of space stations, tiny balconies above the Earth, where life is placed directly against the infinite. Inside the station, there is warmth, pressure, breath. Outside, there is nothing. Vacuum so perfect, it drinks away every drop of water. Radiation sweeps across from the sun, from the galaxy, from ancient stars. Temperatures swing wildly, blistering in sunlight, freezing in shadow. This threshold is sharper than any cliff, any desert, any storm. And yet across the decades, humans have placed small capsules here to see if life can linger in the most hostile stage imaginable. Imagine drifting at 400 kilometers above the planet. Below, the oceans glow in ribbons of blue, clouds swirl in slow spirals, cities wink like scattered fireflies. The station orbits swiftly, every 90 minutes. Day turns to night, night to day. The platform outside the hull swings from heat to cold, from white glare to black silence. Here, tiny panels are bolted into place. They are called expos. They are part of the European Space Agency's exploration program, though other missions bore similar names: Biopan, LDEF, Tanpopo. Each is a little stage where biology is tested against the void. Scientists call it astrobiology. But in truth, It is something softer, more dreamlike. It is a question asked on behalf of every fragile organism that has ever lived. Can we survive beyond home? The earliest experiments were not outside stations, but still above Earth, launched in satellites, carried into orbit for a few days, then returned. In the 1960s, even before humans stepped on the moon, spores of bacteria were carried to space. Some survived. Seeds were carried up and planted back on Earth. Many sprouted. Already the suspicion was growing. Life could endure brief journeys in space. But the real question was deeper. Could it survive direct exposure, without atmosphere, without shielding, with nothing but the emptiness pressing against it? The answer would take decades of careful, daring trials. In the 1990s, European researchers launched a series of missions aboard the Photon satellites, each carrying a biopan, a small capsule that opened its lid in orbit, exposing samples directly to space. Then biopan scientists placed bacteria, lichens, seeds, crustaceans, spores, tardigrades. The lid opened. The samples lay bare to vacuum and radiation. For days they drifted in silence, orbiting the Earth. When the capsule returned, the results astonished. Some seeds still germinated. Some lichens resumed photosynthesis. Some tardigrades, husked in cryptobiosis, revived. The conclusion was almost unsettling. Earth as boundary was not the end of life as endurance. The void itself was survivable, at least for a time. With the construction of the International Space Station, a new possibility appeared. Long-term platforms. Instead of short orbital trips, samples could be exposed for months or years. Thus were born the exposed facilities, mounted on the station's exterior. These were like small windowsills where containers of organisms could be left to face the cosmos. Each exposed mission carried dozens of life forms, bacterial spores to test survivability of Panspermia, seeds to study long-term preservation, fungi and lichens to see if photosynthesis could return after space, tardigrades and rotifers as model survivors of cryptobiosis. Inside their tiny cassettes, these organisms faced raw space for up to 18 months. When brought back inside, many stirred. Some lived, some even reproduced. This was not immortality. Many perished. DNA damage was severe, yet the fact that any lived was enough to reshape our imagination. Japan's space agency, JAX, launched Tampopo, a series of experiments on the Japanese Kibo module's exterior. Tampopo means dandelion, a flower whose seeds drift on the wind. Here dust, microbes, and organic molecules were tested against the cosmos. Could life cling to tiny specks drifting between planets? Could panspermia happen grain by grain, dust by dust? The answer was delicate. Survival was possible, but fragile. The harsher the radiation, the shorter the endurance. Yet even fragile survival suggested possibility. It meant that life might travel not in rockets or capsules, but in microscopic journeys, drifting invisibly across the stars. Why do we keep opening this door? Partly it is science. We need to know how long seeds can be stored, how microbes behave in orbit, how future explorers might grow food or carry ecosystems beyond Earth. But beneath the science is something more tender. These platforms are humanity's first experiments in asking life to share our journey outward. We do not go alone. Carry tardigrades and nematodes, spores and seeds, lichens and algae. They are ambassadors of Earth's resilience, standing on the balcony above the planet. Every time the station door opens, we send out not only data, but hope. What have these experiments taught us? Radiation is the true enemy. Vacuum can be endured, cold can be endured, but cosmic rays slice through DNA like glass shattering. Survival depends on shielding, or on the power to repair damage after revival. Water is both danger and key. Without water, many cells collapse, but with too much water, freezing destroys them. The trick is removal, replacement, and careful pause. Not all life is equal. Complex organisms perish quickly. But simple, cryptobiotic, or dormant forms endure astonishingly long. Time is survivable in fragments. Some can last weeks, months, even years. It's not forever, but long enough to drift between planets or to be stored for interstellar journeys. Life is more portable than we imagined. Not all life, not in all forms, but enough to suggest that biology is not confined. The barriers are not as firm as once believed. For astronauts, the station door marks the difference between safety and peril. For microbes, seeds, and husks, the door is even sharper. Inside, warmth, breath, food, protection. Outside, silence, radiation, and the endless expanse. Yet across the years, across missions and nations, we have dared to set life outside and wait. The experiments whisper back a strange truth. The boundary is thinner than we thought. Its life, though fragile, can stand at the threshold. The outside platforms of space stations are not vast gardens or bustling habitats. They're quiet ledges, exposed balconies. Yet they have shown us something profound. They tell us that life can step outside its home. That the leap from planet to cosmos is not infinite. It is survivable, if only in small, waiting forms. So the balcony above Earth becomes a symbol, a place where fragile creatures meet the void and return to tell the story. The story is always the same. Life is more enduring than it looks. As you rest now, imagine yourself standing at that quiet threshold, the door of the station opening to stars. Inside is warmth, outside is silence. And between them, balanced, waiting, is the secret of survival. When newspapers write about tardigrades, they call them indestructible water bears. When headlines announce spores returning to life after centuries, they say immortal. When seeds sprout after 1,000 years of rest, the world whispers, miracle, but the truth is quieter. They are not superheroes. They are not invincible. They are simply patient. They wait. They endure. They let time slip past in silence, and when it is safe, they breathe again. We humans love heroes. Imagine shields and armor, powers that shrug off fire, radiation, vacuum, or poison. When we see a tardigrade revived after the vacuum of space, we imagine a tiny creature flexing its muscles, wrestling cosmic rays, grinning at the void. But that is not what happened. The tardigrade did nothing. It curled, it dried, it stopped. It let its systems freeze, let its chemistry slow to glass. And then it waited. The miracle was not action, it was stillness. Science calls this cryptobiosis, anabiosis, dormancy, latency. But beneath the technical words is a simple truth. Survival comes not from fighting, but from surrendering. Tardigrade dries itself into a husk, losing nearly all water. Bacterial spore tightens its coat, hiding its DNA within protective walls. A seed hardens, sealing its embryo in armor, drifting for seasons or centuries. None of them resist actively. They yield. They let the environment pass over them like a storm over stone. This is not strength in the usual sense. It is patience beyond measure. When metabolism stops, or nearly stops, time stretches. A human burns through oxygen and sugar every second. Our bodies demand constant fuel, constant flow. We live at speed, in unbroken activity. That is why we cannot last without food, air, or warmth. But organisms in cryptobiosis almost step outside of time. Their chemical reactions slow so much that in practice they nearly stop. It is as if they fold themselves into a page of the book, then close the cover. The story is paused. The ink does not fade. They wait for a hand to open the book again. Patience alone is not enough. These organisms also carry tools, small, quiet tricks that help them endure. DNA repair systems. When radiation breaks the threads of life's code, some microbes can stitch it back together when they wake. Protective sugars like trehalose. These replace water, forming glassy shields around proteins, preventing collapse. Proteins called LEs. Late embryogenesis. Abundant proteins. They act like scaffolds, keeping the cell structure safe during dryness. Antioxidants and pigments. Some shield against radiation, others absorb light. Still others scatter harm. Don't. All these tricks are not about conquering. They are about preserving, but holding a fragile shape steady through the storm. It is like placing a fragile vase inside a padded box, not to make it unbreakable, but to let it wait unharmed until the world is gentler. A superhero fights, resists, conquers. A super patient creature yields, pauses, hides. One burns energy and bursts. The other spends almost none, trusting that time will turn. In this difference lies a lesson. The survivors of cosmic emptiness are not the fastest, strongest, or fiercest. They are the ones willing to wait. They have no pride in action, only humility and endurance. Archaeologists have found seeds buried in tombs, sealed in jars for thousands of years. Some, when planted, still sprouted. Their endurance was not magic, it was patience. The seed did not resist dryness, simply stayed still, its embryo wrapped in quiet, its shell keeping time at bay. In frozen soils of Siberia, seeds tens of thousands of years old have been revived, coaxed into flowering again. The patience of a plant embryo outlasted entire civilizations. Not superheroes, just patient. Bacterial spores have been found to survive for millions of years, buried in salt crystals or amber. When given water, some still grew. Inside, their DNA waited, wrapped in proteins, locked away from decay. In labs, scientists have revived spores from ancient caves, from permafrost, from rocks deep underground. The patience of microbes spans epochs. They are not invincible. Many die, but enough linger to remind us. Life is not quick. Life is patient. Tardigrades get all the fame. They survive boiling, freezing, radiation, vacuum. They do not do so by force. When conditions worsen, they withdraw water, fold into a tiny shriveled husk. Inside, their molecules are held in glass, frozen in silence. When water returns, they unfurl. Not instantly, not triumphantly, but slowly, carefully. They begin to move, to feed, to live again. They are not superheroes in capes. They are monks of patience, masters of pause. Humans are impatient. We measure life in decades, hours, minutes, seconds. We rush, we burn. Our heroes are fast, powerful, dazzling. But the universe belongs not only to the quick, it belongs also to the slow, the waiting, the patient. The microbes and spores and husks teach us something radical. Sometimes survival is not about action, but about stillness. Not about fighting, but about yielding. Not about speed, but about waiting. Perhaps one day, if we travel to other stars, it will not be in heroic leaps, but in slow, patient drifts. Our seeds, our spores, our microbes waiting quietly in the dark until a new sun rises. As you rest now, imagine yourself not as a superhero, but as something simpler. A seed wrapped in quiet, spore hidden in amber. A tardigrade, curled in a husk. No rush, no fight, only patience. The universe may rage outside, the cold, the radiation, the silence, but within there is waiting. Within there is time stretched thin and gentle. Not superheroes, just super patient. The creatures that survive the harshest places are not immortal. They die often, and many never return. But some wait long enough. Long enough for rain to fall, for warmth to return, for doors to open. Their secret is simple. They are not invincible. They are not superheroes. They are just patient. And so, as you drift to sleep tonight, remember, survival does not always mean strength. Sometimes it simply means waiting, quietly, gently, trustingly, until the world becomes kind again. When we think of travelers, we imagine motion. Ships across oceans, rockets through sky, footsteps over mountains. But not all travelers move quickly. Some do not move at all. They are carried by wind, by water, by stone, by stars, spores, seeds, dormant microbes. The million-year travelers of the cosmos, they are not rushing, not conquering, they are waiting. Patient particles of life, drifting in the dark, until a new world offers them a chance to wake. It's inside meteorites that fall to Earth. Scientists have sometimes found organic molecules, traces of amino acids, hints of chemistry that once stirred elsewhere. Some wonder, could entire microbes hide inside these stones? Shielded from radiation by layers of rock, frozen in silence, could they cross the void between planets? It is not impossible. When meteorites are ejected from Mars by great impacts, fragments sail through space for thousands, even millions, of years before falling on Earth. Some carry microscopic pores, shelters from cosmic rays. In laboratories, scientists have tested spores under simulated space. Some survive the vacuum, the radiation, the cold. Not forever, but long enough. Perhaps then, the million-year travelers do not walk, they wait, tucked in stone, hitching rides on the journeys of worlds. Inhale slowly. Imagine yourself curled safely inside a pebble, floating through the dark. Exhale gently. The stars drift past, and time feels endless. Not all travelers need stones. Some are so small, so light that sunlight itself can push them. Interplanetary dust streams through the solar system, a river of motes invisible to the naked eye. Could spores smaller than pollen ride those streams? Could the solar wind or the faint pressure of photons carry them between planets? On Earth, bacteria ride in clouds, lifted kilometers high. Fungal spores drift across oceans. Some scientists have found microbes alive in the stratosphere, far above the ground, where ultraviolet light batters and freezes them. Still, they endure. If they can reach that high, what stops them from reaching higher? From being swept out, carried by sunlight, whispered into interplanetary night? For us, a journey between planets seems impossibly long. To cross light years, unimaginably so. But for spores, seeds, and tardigrades, time is different. In cryptobiosis, the clock almost stops. 100 years may feel like a pause, 1,000 like a nap. As long as their protective shells hold, as long as radiation does not tear their codes apart, they wait. Some will fail, most will fail, but not all. In the vast lottery of space, survival does not require certainty, it requires only persistence. Millions may perish, but one may land where water flows. And then the story begins again. Inhale deeply. Imagine 1,000,000 tiny seeds scattered across eternity. Exhale softly. One lands, one wakes, one grows. This idea has a name, Panspermia, seeds everywhere. It does not mean that life certainly began elsewhere and arrived here. It does not claim certainty, but it whispers of possibility. That Earth, with all its oceans and forests, might be the child of a seed carried from afar, or that Earth, in turn, is scattering its own children. Microbes lifted into space, spores clinging to spacecraft, seeds launched accidentally into forever. Life, then, is not confined to one planet. It is a wanderer, a passenger, a million-year traveler. These travelers are not superheroes. They are fragile, vulnerable. A spore's shell is thin. A seed's heart is soft. Tardigrade's husk is delicate. Most will never survive. But patience changes the odds. Stillness allows them to wait out not just seasons, but epochs. And across billions of chances, across countless failures, a few may endure. This is how life works, not by certainty, but by persistence, by scattering, by drifting, by waiting. Breathe in. Imagine your life as part of a vast, slow journey. Breathe out. Imagine the patience of seeds, spores, husks, and hushed travelers. We dream of rockets, engines, warp drives. We imagine conquering space and speed. But perhaps in the long arc of the cosmos, we too will travel like seeds. Frozen embryos drifting, genetic libraries stored in crystal, humans in hibernation, learning the patience of tardigrades. Maybe our true future among the stars is not in rushing, but in waiting. Not in conquering space like superheroes, but in joining the quiet caravan of million-year travelers. As you drift towards sleep, imagine this. Across the galaxy, there are seeds floating, spores locked in stone, dust-carrying embryos. Silent passengers of time. Most will fall into nothingness, but a few will land on gentle ground. A few will wake. A few will bloom into forests, oceans, skies. Perhaps Earth itself was once only a seed. Perhaps you are part of a story begun by a traveler from a star long gone. It's not superheroes. It's just patient. Not conquerors, just survivors. Not fast, just enduring. The million-year travelers drift on. As you rest tonight, you drift with them. The journey is almost over now. You have wandered with me through airless voids and frozen husks, through brine shrimp cysts and nematode canisters, through seeds that wait for centuries and spores that drift like dust among the stars. You have met the patient ones, those who travel not with haste, but with silence. The tardigrade rolled tight into its ton. The rotifer curled like a ribbon of memory. The nematode cradled in its capsule. The brine shrimp cyst, the Daphnia egg, the resting seed. Not superheroes, but simply survivors. Not conquerors, but custodians of patience. And what have they shown us? Life does not always win by strength or speed. Life sometimes wins by waiting, by hiding, by trusting time to bend, eventually toward renewal. Above you, the stars are watching. They have burned for billions of years, and they will burn for billions more. Compared to them, we are fragile. Yet compared to us, even the smallest spore seems eternal. This is the paradox of life, fragile and enduring all at once. A single ultraviolet photon can strike and shatter, and yet somehow across deserts, glaciers, oceans, and space itself, life still finds ways to linger. As you close your eyes, imagine yourself among the million-year travelers, not rushing, not racing, simply drifting, gently, in stillness. Imagine yourself curled inside a safe shell, light as dust, timeless as stone. For a moment, you belong to no clock, no calendar, only to the quiet patience of the cosmos. Inhale softly. Let your breath expand, like the slow unfurling of a seed. Exhale slowly. Let it rest, as though sinking into dark, fertile soil. Do you see them now? Tardigrades on spacecraft windows, brine shrimp cysts curled in dusty flasks, rotifers ribboning back to life when the water returns, worms that endured the fall of Columbia, survivors of both flame and sky, spores locked in stone, waiting perhaps 1,000 centuries. They are not myths. They are not immortal. They are simply proof that endurance comes in many forms. And as you drift into rest, know this. You too are part of that same story. Your body is made of cells that repair, adapt, and endure. Your heart beats with rhythms that echo the patience of the ancient ones. Your breath carries the same molecules once carried through stars, through dust, through oceans, through time itself. The stars are watching. The spores are waiting. The million-year travelers drift. Tonight you join them, resting, breathing, patient in your own quiet voyage. Even in the darkest places, life endures. Even in the longest journeys, rest is part of survival. Even in the silence of space, there is a whisper. Wait, and you will wake again. So now, close your eyes. Let the stars keep their watch. You have traveled far enough for tonight. Sleep now, beneath the watching stars.